In this video, we will learn about the cell cycle regulation mechanism. To understand the cell cycle regulation mechanism, first we should know the importance of regulation. Why cell needs regulatory mechanism for its cell division. That is very important to learn in this video. So, first, let's focus on the importance of the regulation for a cell division. Since we know that the cell division it replaces the damaged cells, or it helps organism to grow in size. Also, it involves in production of reproductive cells, which transmits that genetic information to the next generations. So all these events are very important. That means when organism needs, or when organism needs to replace its damaged cells, then only the cell has to be divided. When the old cells are present, only those old cells only are to be replaced in the organism. And during the growth of organism also, number of cells, or the number of cells present or required for the organism are also must be known for the cell division. And then finally the reproductive cells formation. There we know that the genetic stability is more important, because this information will be carried forward to the next generation of daughter cells. Therefore, that genetic information must not have any errors. So to organize all these things, or to coordinate all these things to happen inside of the during the cell division, so there must be a regulatory mechanism to control all these. So we know that there are two phases in a cell division. One is the mitotic phase, another one is the interphase, and we know that there are different stages again in interphase, like G1 phase, S phase, and G2 phase. And in G1 phase, the cell gets prepared for synthesis of DNA in the synthesis phase, which is the next phase after the G1 phase. Then in S phase, the DNA gets duplicated to distribute to the daughter cells during the mitotic division. Then in G2 phase, the cell gets prepared for mitotic division. And very interestingly, there are a few proteins which coordinate all these processes, or all these events in cell division. So there are two proteins which plays an important role in cell division. They are called cyclin proteins and kinases proteins. So these proteins, cyclin dependent kinases needs cyclin proteins. So cyclin proteins, with the help of this cyclin dependent kinases, they phosphorylate the proteins which play important role in preparing the cell for S phase, or synthesizing DNA in the S phase, or in preparing the cell for a mitotic phase during the G2 phase. So there are some proteins which are required, which must become active during these specific phases. So these cyclins, with cyclin dependent kinases, they phosphorylate these proteins, so that which are in inactive condition becomes active, and perform the respective functions in this G1 phase, S phase, and the G2 phase respectively. Okay. So coming to the cyclins, why they named these proteins as cyclins? Because these proteins levels gradually increases and declines, and also these cyclins, we see that different cyclins are present, like cyclin A, B, E, D. And these cyclins, they present, that means they increase and decrease, or decline in a specific time periods, or in the specific phases of this interface. Like when we take the when we take the cyclin D, so the alphabetical naming of these cyclins are also, it's the order of their discovery. So coming to these cyclins, as I said, no, these proteins, they gets upregulated and downregulated during the this cell division. That means these proteins are synthesized by the genetic material of the cell, and after their synthesis, so these proteins undergo the proteolysis. That's why their concentrations gets increased and decrease during the cell division, okay, in the interphase. Okay, coming to the cyclin D, this cyclin D is actually the protein which is synthesized in response to the growth factors. Therefore, so during the G1 phase, so these growth factors, when they're present in the vicinity of the cell, so these growth factors, they activate the genes. These genes, they synthesizes the cyclin D protein, and very interestingly, this cyclin protein D, so the levels of cyclin D protein, if we see, so their synthesis starts in the G1 phase when the cyclin D protein synthesis starts, so that cell gets ready for the this interface, so that no cyclin D levels, when we see, so the cyclin D levels are present throughout the interface, unlike other cyclines, so whose concentration actually is limited to a specific phases only. So here coming to this cyclin D, whose level starts increasing in the G1 phase, and their levels of that means G1 levels, they are present throughout the this interphase. So why these cyclin D levels are present throughout, or why cyclin D remains throughout the interface is very important to know, because the growth factors. So as long as the growth factors are available for the cell, so the start synthesizing the cyclin uh, D proteins. So that's why. So since the growth factors may present around the cell during the entire phase, so therefore the cell produces the cyclin D proteins. Okay. Then next one is S phase. So when the cell enters from G1 to S phase, there must be one cyclin protein which is very important, that is called a cyclin E protein. So actually, this cyclin E, the synthesis of cyclin E starts between the G1 to S phase. So that means it exactly when the G1 when the cell crosses from G1 to S phase. So at that point, so the cyclin E synthesis starts in the cell. So the cyclin E levels, we can see that no, these levels, the cyclin E proteins are very important to prepare the cell for the S phase. To prepare the cell for the S phase. Very interestingly, the cyclin E proteins, so these cyclin E proteins, with the help of the CDKs, so they phosphorylate proteins. So these phosphorylated proteins, they activate the certain genes in the genome. So these genes produces proteins which are required for the DNA replication. So the proteins which are required for the DNA replication are synthesized in response to the cyclin E and the CDKs. Okay, that is about the S phase. So after S phase, the cell enters into the G2 phase. When cell reaches to G2 phase, so we can see that no, there is another cyclin proteins, we call them cyclin A proteins. So the cyclin A proteins, they start synthesizing actually in the S phase itself, and their levels reaches to peak at the G2 phase. Okay, cyclin this cyclin B proteins. So sorry, cyclin yeah, A proteins. So after this, so the cell enters into the mitotic phase. So before it reaching to the mitotic phase, so this cyclin, what I'm saying now, cyclin A proteins. So the cyclin A proteins are required for the cell uh, to make the proteins which are required for the cell to exit from G2 to the M phase. So some proteins which are required for the cell to exit from G2 to M phase. So those proteins are synthesized by the cyclin A. Those proteins are activated by the cyclin A and CDKs. Okay, then the other proteins are called cyclin B proteins. The cyclin B proteins are required for the cell during the mitotic phase, not during the interphase. These proteins are required during the mitotic phase, and whose synthesis starts even in the S phase itself, but their levels reaches to that means the protein accumulates over a period of time, and their concentration reaches to peak at M phase, and these cyclin B proteins are required for the cell to complete its mitotic phase. We know that there are some proteins which are very important, or some enzymes which are important in mitotic phase, and those enzymes or proteins are phosphorylated by these cyclin proteins and the CDKs. Okay, so this is about the cell cycle regulation, and the other important thing that we have to understand, that no, see, the cyclins and CDKs, they don't directly participate in the this kind of what interface, or they don't directly participate in the mitotic phase. That means they don't synthesize DNA, they don't help the cell to grow, they don't help the cell to complete its prophase, metaphase, anaphase, telophase directly, but they actually know they activate the other downstream proteins. So those downstream proteins, they participate in these reactions. That's why we say that the cyclins and the CDKs, we call them as regulators, because they're like our principals. So who doesn't take classes for all the student, but the principal regulates the student teachers. Therefore, the teachers, they take classes for the students. So this is how the cell cycle mechanism works.